This basically gives all the biological functions or genes grouped by, let me group this. I'm going to size these boxes by the number of genes associated with these particular functions that were in my gene list. So as far as abdominal neoplasia, which is obviously very important in the development of epithelial cancers, I had 390 genes that showed up out of my list of 960. Not only that, but based on the direction of the expression of those genes, that this thing is definitely more active. But it makes sense that when you look at things that are down, like cell death, that makes sense. Right? And also, as we look around here, you look at this very dark blue. So this is predicted to be inhibited. Organization of cytoskeleton. What I find in these advanced cancers is when they go metastatic, they kind of start breaking down their cytoskeleton and they actually start detaching. That's kind of a sign of metastasis. You can see it. And again, we can go to any of these things and actually download all of these genes. So here are all my genes associated with organism of cytoplasm. In this column, this shows me the evidence that when chemokine 10 goes up, it increases organization of cytoplasm. And here is all the evidence, and I can click on this, and it'll get me to the paper that actually proves that or shows that. But as you look, there's more evidence for decrease than increase, and that's where it gets that confidence call. It's basically just an F value. The statistics actually behind all of this, I know it looks very impressive, but the statistics are incredibly easy. It's basically, given a limited number of things to choose from, what are the odds that I get all of this specific subset? It's an F test. Okay. Click here. We can actually go to tox functions and look at things that might be associated with that. I'm going to tell you right now that there's obviously a lot of people doing cancer research. So, a lot of software is kind of geared towards cancer, that it's kind of overrepresented with a cancer. Usually when I see cancer, it just means there's either the cells are dividing or differentiating, or it's usually a differentiation state difference. Okay, so disease and functions, and again, we can look here and look at things that are more inhibited versus more active. Let me see. Oh, let me go back to disease biofunctions. Formation of cellular protrusions is my most inhibited biological function, which is interesting. Oh, wait, I'm on the wrong one, sorry. Oh, there it is. Synthesis of protein is down. Senescence of cells is down, which you would expect with cancer. And then we look at the opposite, proliferation of cells are on fire. These cells are dividing. Not only are they dividing, but they're detaching from their neighbors. And I know this by looking at this data.
So what's the green and red arrows there?
This would be that this goes down in stage 3 if it's a green arrow, and red would be it increases with it. Okay. And again, we can get all of these particular, molecules out associated with any of these things. I can make a subset and actually analyze just maybe a subset and do the same thing, except say, I just want to look at the 200 genes that are associated with like the cytoskeleton. And I can do my analysis on that. It's pretty easy to subdivide all this stuff. Let's see. Let's go to, so you got disease and function, canonical pathways. Here's what I think sets IPA apart, is that what it will do is it'll group genes based on just any relationship whatsoever in the literature. It isn't dependent. These genes don't have to be in the same pathway. They don't necessarily have to be in the same biological function. If there's evidence somewhere in the literature of these things interacting together, it'll show that. And so what it'll do is it'll build these networks of all these interactions that aren't necessarily canonical. These aren't your typical Go pathways. And I'm going to tell you right now, most Go pathways, your standard pathways that people have developed, they were developed, some of them are like 10, 20 years old. They've been developed in all kinds of tissues with different organisms. They're kind of like your cliff notes for it. In my opinion, these are actually your true signaling networks, these networks. We can pick this out. Can everybody see that? Everybody see what's in the middle there? Yes, yeah, that's an estrogen receptor again. Look at all the things it's interacting with. Again, it's going down, but a lot of these things are going up. We can actually color these relationships. If the software knows what this relationship is, it'll give that. So I can go overlay, and I can go to map activity predictor. I'll say start prediction. Oh. Hey. So you can see, so if it's yellow, it means that given that this source molecule is going down, it's normally inducing this, and this is going up. That doesn't jive with the relationship here. It's basically kind of showing that broken kind of signal that I was telling you about. Some of them, but most of them, they are. You know, when I talk with the high school students, we talk about kind of godfathers. Now, when you're actually analyzing your data sets, you're not interested in the low-level thug, you know, selling drugs on the corner. You want to bust the, you know, the big chief, you know, the big godfather. These things, when you actually look in the data, what you're looking for are things like this that interact with lots of things in your gene list. And we're going to see that in the upstream regulators. But I can overlay all kinds of things here. I can see if any of these are associated with a particular pathway. 3 of these are in row signaling. And I can mark those there if I wanted to. If I click on here, it'll light up the ones that are associated with row signaling. We can also do a grow function. So basically what I can do is I can take any of these networks and I can overlay all my pathway, my biological function information, anything I want. Okay. Now for the godfathers.